But my name is Gideon Mordecai. I'm a research associate here at the IOF. I study salmon health and disease, and I've been tasked with introducing today's speaker. But first, we'd like to acknowledge that the land on which the Institute for the Oceans and Fisheries is broadcasting this seminar from is the traditional, ancestral, and unceded territory of the Musqueam people. So today's speaker is Dr. Sandra Binning. Dr. Binning is an associate professor at the University of Montreal and Canada Research Chair in the eco-evolution of host-parasite interactions. Sandra is originally from Montreal, where she did her master's degree at McGill, but she earned her PhD at the Australian National University in Canberra, studying how the shape and function of coral reef fish change with different wave and water conditions. Sandra then moved to the University of Neuchâtel in Switzerland for a postdoc, continuing her research on the Great Barrier Reef. So Switzerland's a landlocked country far from any coral reefs, but the Swiss franc goes a long way, and that's where she explored cleaning mutualisms and how parasites affect fish behavior and cognition. And Sandra returned to Canada in 2018 to join the University of Montreal. So I'm really looking forward to this presentation of how fish will tolerate pathogens under heat waves and climate change. I think it's very topical. So welcome, Dr. Binning, and I'll hand it over to you.
Thanks a lot, Gideon. I appreciate that. Can you hear me okay? Is this a good volume? Great. Okay. Thanks a lot for the introduction and also for the invitation to come and give this seminar to your institute today. I have to say it was really the kick in the butt I needed to update my seminar talk. Catherine, when we were discussing what I should talk, she said, I don't have to talk about marine stuff, but I do have to talk about recent stuff. And that got me thinking, okay, I think at this point in my career, I can officially say that my PhD work no longer counts as recent. And so I've decided to revamp my seminar and really focus on some of the work I've been doing with my students since I started my position at the University of Montreal in 2018. So I'm not going to be talking about my PhD stuff, probably for the first time in a very long time. So you can thank me for that. That does mean, though, that my background, even though it's more on marine systems and coral reef systems specifically, this talk will really focus mostly on freshwater fishes because I am based in Quebec, and in Quebec, we do have a lot of lakes and freshwater systems in general. And let me give you a few stats for those of the more salty water focused people here on why freshwater systems are really important. First, they cover kind of a small percentage of the Earth's surface, but they really punch above their weight in terms of biodiversity. They contain almost 10% of free living animals, about 30% of known vertebrates. And if we're, I mean, not that this is a competition between marine systems and freshwater systems, but if we are doing a comparison, they actually host, they're home to more active oxygen fishes than marine systems, about 15,150 species in freshwater systems compared to about 14,700 species in marine systems. So again, as far as biodiversity goes, they're really, really important critical ecosystems. And the story that I'm going to be telling really starts over on my side of the country in the Laurentian area, just north of Montreal, where I'm based, by these beautiful lakes. This is Lake Crush. It's one of the lakes we work on in the fall. It's a really lovely place. And I guess for most people, not even just in Quebec, I mean, pretty much everywhere around the world, there's lots of memories we might have as kids growing up, spending our summers near a lake like this by an ocean, where we go and cool off from the hot summer heats from these increasing heat wave days that we're getting as our days are warming. And these thermal refuges that freshwater systems offer us are really becoming more and more critical resources, more valuable for us as a resource in general. But for fishes, it's kind of a different story for other organisms that live in these lakes as well. We might go to a lake to cool down, but for the species that are living in those areas, these increases in temperature that they're experiencing are really becoming a big problem for them in their sort of lakeside paradise. And I think over the last 10 or 15 years, maybe, and especially more recently in the last 5 years, there's been a growing realization, I think, that freshwater systems are in some ways really uniquely threatened by different stressors posed by climate change. And I think there's increasing research effort that we're seeing, and it's really dedicated to trying to understand the consequences of warming waters for freshwater species and how given species might or might not be able to tolerate these changes in terms of different aspects of their performance. And this is just a selection of some of the recent papers that have come out on the topic, really exploring specific threats posed by global warming or by different aspects of climate change to aquatic species, but freshwater species in particular. And when we're interested, I'm an empirical biologist. When I go and I'm interested in studying how species are affected by things like temperature increases or any aspect of global change, what I typically do, and I think what a lot of people typically do who work on empirical systems, is that we may collect them from the wild, we bring them back to the lab, we subject them to different tests to assess how well they cope with a given stressor. That's kind of mostly what we do our approach as empirical biologists. But when we take an organism, especially from the wild, bring it back into the lab to measure its performance, we're making a few pretty key and critical assumptions. We're assuming that our animals are healthy, that the performance that we're measuring is maximal, and that any behavior we're observing is adaptive. And these assumptions are actually pretty problematic when we take a step back and think about what an organism is actually dealing with inside of it. And that's because in reality, wild organisms aren't just exposed to stressors like temperature, they also have parasites. Most of them are too small for us to see and notice. And even though I think most biologists would rather pretend that it's not true, this idea has pretty profound implications for how we study and measure any aspect of performance or function in our system of interest. And that's because parasites they're kind of everywhere, right? Like some estimates put almost 40% of known species as having at least a parasitic stage at some point in their life cycle. And again, that's just where we thought to look at them. Most parasites or most species, we haven't really fully described the parasite fauna that infect them. So we know very little about the parasitic organisms that infect most species. Most importantly, parasites infect almost all plants and animals. And at any given time, most wild organisms are infected with or at risk of being infected with something. So no matter what species you work on, some aspect of their biology has adapted in response to the risk of parasite infection. And understanding something about the effects that parasites can have on their hosts can tell something really fundamental about our species of interest. Unfortunately, the impact of many parasites and diseases on organismal function and performance is relatively unexplored, especially in the context of other stressors like warming or hypoxia, toxin exposure, you name it. There really isn't a whole lot of work looking at how these multiple stressors act synergistically or additively to affect the host. But how big is this problem? If most wild organisms have parasites, well, how often are they or not considered in research? During the height of the pandemic, we couldn't access our beautiful field station, which I put in the picture here just for fun. And so we decided to tackle this question through a systematic review of the literature, where we quantified if and how parasites were considered in studies of behavior and physiological performance in wild animals. And again, this was a pandemic, and so our fieldwork, instead of taking place in our beautiful field station, was more or less in the bottom of our basements in the dark. But again, it was, I think, a productive time nonetheless. And so what we did was we screened 680 different articles published in a variety of different journals that publish different aspects of animal performance. And we found that only 22% of the articles that we screened had any form of parasite awareness, any mention of parasites in any of the wild organisms that were studied. And of these 149 articles, less than a quarter of them controlled for or included a metric infection in their analyses. The majority, even if they mentioned that there maybe were parasites that they were found in the species they were studying, took no further action to kind of incorporate them or look at them in their analyses. And I think that's pretty unfortunate. I think it's safe to say that parasites are, again, pretty often ignored by biologists, and that oversight can have really important far-reaching implications for biologists, especially those doing experimental work, because infection can have really important and profound and sometimes counterintuitive impacts on their hosts. And ignoring them can lead to bias or misinterpretation, sometimes even erroneous conclusions. There's actually a really good paper by Juan Timmy and Robert Poulet that specifically focus on the issues that one might have in fish ecology if you're ignoring parasites, you're not taking those infections into account. And I encourage everyone to have a look at it. It's really, really interesting. But more than that, I think there's also an overlooked opportunity or misunderstood opportunity to understand some of the really key drivers of phenotypic change and sources of biologically relevant variation within a population, which I think is really important if we're trying to understand which species or individuals are more likely to be resilient to different aspects of global change. And I think that that's even more important as we're leaning towards a world that's potentially getting more warm and also potentially more wormy. That is kind of an open question, though. Is the world actually getting wormier and warmer? there's a bit of evidence that suggests that disease incidence is increasing, or at least likely to increase as a result of warming. And there's an excellent paper by Sonia Altsier on infectious diseases and climate change, and different examples about how there's increasing incidences of these interactions as the world gets warmer. But a lot of other research actually suggests the opposite. There's some really great work by Chelsea Wood, who's looking at these questions, and she's actually found by going through some museum specimens that there's evidence that actually some types of parasite burdens are decreasing in fishes over time as the climates warm. Basically, we need more research. There's just not enough information to really be able to effectively evaluate these questions. We need more research, especially experimental, to better answer whether or not parasites are impacting host performance and whether that's likely to get worse as climates warm. And that's where I think my lab is trying to tackle some of these questions. The way we're doing that is by studying the pumpkin seed sunfish. It's this beautiful freshwater fish right here. I have to say, as someone who has worked a lot on coral reef fishes, they're pretty special fish, even though they are freshwater. They're very common freshwater fish native to Eastern North America. We have lots of them in our lakes and rivers around Quebec where I work. They're small, they're really curious, easy to collect and keep in captivity. They also live across a really broad temperature gradient. They actually are kind of concern in some areas because of their invasion potential. They're invasive in parts of Southern Europe, where again, they live in these waters that are already much warmer than what we're experiencing here in Quebec. They also have really interesting behaviors. They have male parental care. He has a little daddy on top of his little nest over here. So there's some cool reproductive behaviors that we can look at. We're starting to get into that more. I'm not going to present any of that work today, unfortunately. But I think for our perspectives of today, at least, the interesting thing is that they're host to a really wide variety of different types of parasites. And I'll talk a little bit about some of them now. One type of parasite that we commonly see in our fish is black spot disease. This is a disease that's caused by parasitic worms called trematodes. There's many different species that cause these diseases, and they infect many different types of fishes, not just our pumpkin seed fish. All of these sort of parasites that cause the black spot disease have the same generalized life cycle. It's a complex life cycle, so the parasite needs to infect 3 different hosts in order to finish its reproductive cycle. They start off their life in a freshwater snail. The snails release these free-swimming cercaria that kind of go and find a host, like pumpkin seed sunfish. They penetrate through the skin, and what happens is that as they penetrate, the host's immune defenses form this melanized cyst around the parasite, and you get these little black bumps that you can see kind of here that, again, melanize and form this really kind of rugose surface on the fins, but across the body surfaces of the fish as well. What's interesting about that for us is that it means that we can go in the water on these transects. We've got a bunch of transects that we establish in our lake systems, and we can actually pretty quickly identify individuals that are infected or not. So we can non-invasively get an estimate of parasite prevalence just by looking to see which individuals have these black spots or don't. That's a really powerful technique that we can use. This parasite finishes its life cycle when the sunfish get eaten by a piscivorous bird, or in some cases mammals as well. Mostly in our systems, it's kingfishers or loons or herons that are the final hosts for these parasites. And then the life cycle kind of continues from there. The other kind of trematode that we have is a little bit similar, but a little bit different in some ways, is something called the yellow grub. It's, again, got the same generalized life cycle, starting from a snail going into our sunfish. But in this case, these grubs, I mean, they're not small and kind of, tiny little bumps. They're really big, chunky things. They're a few millimeters long, each of them. You can see them when they kind of penetrate through in the fins. In our systems, we tend to get them mostly around the gills and also in the heart, around the heart of our fish too. So they really do kind of find their way and kind of make these big, once you pull them out, these big holes inside of the fish where they've encysted. And again, final host for these guys is a heron. And so when the fish gets eaten by a heron, the parasites can complete their life cycle in the heron. Last but not least, we also have bass tapeworm. Those are caused by cestodes. The life cycle is a little bit different. Here we're starting in a copepod. And what happens then is the sunfish will then eat an infected copepod. The copepod the parasite comes out of the copepod, it penetrates through the digestive tract of the fish, And in our systems tends to find residence in a lot of the organs like liver, spleen, sometimes the gonads. But in our fish, we really see it mostly in the liver. And this is just a little picture here of a liver, what is supposed to be a liver. There's a lot of these little worms. These little white things are worms that are kind of really kind of inside it, form this kind of tight mass of where it's difficult sometimes to even distinguish between the fish tissue and the worm tissue. So they really can cause a fair bit of damage to the internal organs of the infected host. And this parasite completes its life cycle when our sunfish gets eaten by a piscivorous fish like a largemouth bass or smallmouth bass. And we're really lucky at the University of Montreal because we have this really nice system available in our backyard. We have access to a field station called the Station de Biologie des Laurentides. It's about an hour and a half away from our main campus. And since about 2019, our team's been surveying sites across a bunch of different lakes. These are 5 lakes that I have here, where we really, they're all within walking distance from the station. And we're able to, again, through our snorkeling transects, really get an idea about the natural gradient of black spot infection prevalence across these different lakes on our different sunfish. Now, one of my students, Julianne, did a study across 12 different lakes in the system to try to find predictors of black spot prevalence across the fish communities. Why is it that we have some lakes where we have basically no infection and others where we have almost 100% of our fish are infected? I'm not going to have time to get into that today, but I encourage you to check out her publication if you're interested in predictors of disease kind of across ecosystems. It's really interesting. But what's important for this talk, at least, is that we have a gradient of infection both within and across lakes. So not only do our lakes differ in infection prevalence, but they also, and the neat thing too about this is that because these lakes are all part of the same drainage basin, they have similar water chemistries and temperature profiles. So despite being very different in terms of parasite loads, they actually are experiencing similar abiotic conditions. So this is just some data from our temperature loggers. We put them in the lake. We try to get them in early mid-May and pull them out sometime before the ice forms in November. And again, you can see that the fluctuations are really similar across these 3 main lakes, where again, we have a pretty strong gradient of different infection from Lake Triton that doesn't have any of these trematodes or cystodes, all the way to Lake Cromwell, which really has super, super high prevalences, somewhere up to 70% of the fish at any given period of time. But again, as I was about to say, within a lake, we also get a really neat gradient of infection. So even fish from Lake Cromwell, this kind of high prevalence infection lake, we can actually get individuals that have barely any infection at all the way to guys like this that are, looking pretty rough, where they have somewhere upwards of 1,000 to 2,000, even 3,000 black spots and other things going on inside them as well. So, we're trying to explore a little bit within that lake. Why is it we're getting these hotspots of infection, hotspots of risk? How are these hotspots of risk within the lake being mediated by different environmental conditions? I don't have answers to that question either today, but that is something that we're super curious about. So we can use this model system to ask questions about the way that parasites contribute to phenotypic variation in the hosts, which again, I think is important to consider in the context of global change and understanding the impacts of warming on individuals and populations. And understanding more broadly about how parasites contribute to phenotypic variation in their hosts is something that I spent a little bit of time thinking about. And my colleague, Lauren Nadler, who's based at the University of Southampton in the UK, we co-edited a joint special feature for the Journal of Animal Ecology, Functional Ecology, in 2023 called Mechanisms and Consequences of Infection-Induced Phenotypes, which again, please check out if you want some more information. There's a bunch of cool studies in there, not just on fishes, but across a broad range of taxa. But for the rest of the talk, I want to give some examples from the research on the pumpkin seed sunfish system that we've been doing since about 2019 to look at basically the way that these 3 kind of worms that I talked about contribute to modifying the morphology, behavior, and physiology of their hosts and the consequences of those changes on host performance. So first, we'll start with changes to host morphology. I mentioned earlier that infection with black spots causes the surface of the fish to go from really nice and smooth and streamlined to kind of rough and bumpy because they create these little cysts that you can see with your naked eye, but you could also feel if you pick up a fish, you really can feel the sort of bumpiness across the surfaces. And you can just see some pictures here that I think illustrate that. And my PhD student, Jeremy, really wanted to know whether this changed the surface roughness or the streamlining potentially of the fish has an impact on their swimming performance. And he tested this by comparing the swimming performance in 2 groups. One were these wild-caught fish that were naturally co-infected with these 3 types of parasites. And in the other group, he actually gave them a deworming treatment called praziquantel. For those of you who have dogs or cats, you've probably given them some of these pills to deworm them to get rid of their tapeworms inside. We can give it to fish as well and get rid of any of the tapeworms that they might have in their bodies. So what we can do is really treat the fish for some types of worms, get rid of those worms, and then just look and see, well, what if you have a fish that doesn't have any of these other worms, but only has these bumpy parasites on the surface? Because even if the parasites aren't metabolically active or are dead, it's still having an impact on the morphology. And so what we do, we can look at aerobic swimming performance in a swim tunnel. For those of you who have never seen a swim tunnel before, it's basically just a treadmill for fish. We put the fish into this chamber. It's a sealed chamber. We force them to swim at different speeds, just like you're forced to run at speeds when you're on a treadmill. And then what we can do is look and see how long a fish or what is able to swim, or how like what maximum speed, what critical swimming speed it achieves. So what's the sort of fastest it can swim in these chambers in these 2 different conditions. And what Jeremy found was that the fish that have more assists tend to swim slower. So this is a graph of the critical swimming speed, our measure of performance in our swim tunnels. And even though there's no difference in the swimming performance between our treated and our untreated group, what we did find is that, again, the more worms that you have, the more black spots you have, the slower you end up swimming. And what this suggests is that this decrease in swimming performance that we find is mostly due to this morphological change in the streamlining that occurs when the cysts are present in high numbers. There's no difference whether you have live worms or not. It really seems to be related to the number of these black spots that you have on your surfaces. It really seems to be messing with your rugosity, your streamlining. So parasites can impact host morphology with consequences for their swimming performance. But what about behavior? There's a lot of studies that have shown that infected individuals have different behavior than their uninfected conspecifics, but it's really complicated. It can be difficult to attribute the cause and effect in these situations unless you manipulate infection, you do experimental infection, which can be really challenging. It's hard to experiment to infect individuals in the lab. But what Marianne did, one of my master's students, she tried a really neat experiment where actually she did want to do an experimental infection, but rather than do it in the lab, she actually did it in these cages that we put out in a semi-natural environment. We put them out in the lake and get our fish infected in nature, and then look and see at the consequences of behavior. So what she did was she collected fish at the start of the summer. They were about a year-old-ish fish at the start of the summer. She gave them a praziquantel treatment to deworm them, just like what Jeremy did, and then measured exploration, boldness, and activity behaviors twice initially in the lab. She put the fish out in these experimental infection cages in our lake, around different areas of the lake, for about 5 weeks, and then collected those fish after that time where they had a chance to get naturally infected in the wild, and then remeasured their exploration, boldness, and activity twice after infection. And again, I think the cool thing about this experiment is it really allowed us to compare an individual's initial behaviors with their behaviors following this infection, and kind of really relate the two each other. And so this is a little bit, just give you some examples of what her tests look like. So she had this boldness test where she looked at the latency of the fish to exit from a refuge, or this sort of like safe side of the tank. So you'll see a little fish swimming around here in the top corner. And then eventually, after a little while, it sort of explores around, decides that it's not too scary to leave or to try to, like I said, these are curious fish. And then it emerges from this sort of refuge area. And we also looked at exploration as a percentage of a surface area covered in this open field test here, and the distance traveled as a measure of activity. So again, just the pretty standard measures of animal behavior. We recorded them using LollyTrack, this automatic tracking software, and then we were able to, again, compare individual differences before and after this experimental infection. And this is a complicated experimental design, and so she used this Bayesian mixed effect model approach. When she found that fish activity did seem to decrease after fish had been infected, specifically after infection with black spot disease. Black spot infection really seemed to be negatively related to individual activity. So infection really seemed to reduce these overall movement behaviors in the species. But that begs the question, well, does that make them more susceptible to predation? Remember, these fish, for the parasites complete the life cycle, they require the fish to be consumed by a piscivorous bird or a fish. And so is there any consequences of this change in behavior on susceptibility to infection? Looking at predation risk in the lab with live predators and prey is really challenging, both logistically, but also from an ethical perspective. But what we can do is simulate a predator attack and then measure the fish's responses to the simulated attack. And when fish are startled by a predator, most species perform a characteristic C start, where they kind of bend their body into a C shape and recoil quickly to achieve really high accelerations away from a potential threat. And so this fish here, I'll just try to show my little video again. This fish here performs a pretty characteristic C start, where I hope you were able to see that. I'll play it again if I can. Let's see if I can play it again. So there's something that falls in the top corner, and then it does this really, really quick burst away from the little object that we dropped. And that's sort of what you should be doing if you're getting attacked by an aerial predator. And so what my student wanted to know is, well, are you able to escape in the sort of same, are you as responsive to this simulated attack when you have a lot of worms or not? And so what she found was that responsiveness to the simulated aerial attack actually decreases when the fish are infected with more tapeworms. So this again is sort of a similar video as I showed before. In this video, you'll see an example of a slightly different response. So in this case, the stimulus drops, but the fish doesn't do anything. And what she found again is this responsiveness, whether or not you actually react in the face of a danger from an aerial predator, or any type of predator really. Really seems to decrease the more of these tapeworms that you have. And this is a really important result, because this response behavior, if you're thinking about it ecologically, is a really important determinant of survivor. Like, yes, you might be interested in looking, when we did look at other kinematics like speed and acceleration, you could be the fastest fish in the lake. If you're actually not responding to a threat, then it doesn't matter how fast you are, you're still going to potentially get predated on. So again, this seems to be a pretty ecologically important response, or lack of response. But of course, a lot of the behavioral changes we see in hosts stem from changes in the physiological processes that occur inside of the host. And so we also wanted to look and see, in my lab, we do a lot of work in ecophysiology, trying to understand how parasites contribute to changes in physiological processes inside their hosts. Unfortunately for the sunfish, the cestodes that I mentioned, these tapeworms, can cause a lot of damage to the livers. I showed you one example of these infected livers before, but I'll just give you a better kind of example of the gradients that we see. So these are livers that come from Lake Triton, which is our healthy, uninfected lake. These are the way livers should look. They're very nicely shaped. They're nice and red, pinky. They're kind of healthy looking. When we compare that with two of our other lakes, Cromwell and Long, that do have a lot of these cestodes, you can see that we've got some of these livers that are not looking nearly as healthy. They have a lot of infection. It's not always a pretty sight. And my students, Sophia and Vincent, have looked at differences in the activity of different mitochondrial enzymes in the tissues of the infected fish. And they found some pretty cool stuff in these two infected lakes, Lake Long and Lake Cromwell. They found a positive relationship between cestode density and lactate dehydrogenase activity. So this is an enzyme that's responsible for or involved in anaerobic metabolism. And so what it suggests is that in these two infected populations, there seems to be this reliance on anaerobic metabolism when parasite burden is high. So again, there might be differences in terms of the ways that animals are getting energy depending on how, and producing energy depending on how infected they are. How does this scale up when we look at the whole organism level? Well, we've also explored how infection correlates with different metrics of whole organism metabolism, including maximum oxygen uptake. And these relationships can be pretty variable. The trends are not always super clear, but we do have evidence that both these internal parasites like these cestodes, but also black spots, are related to decreases in maximum metabolic rate and a reduced aerobic scope in our infected fishes. And again, that seems to be consistent with this idea that infected fish have this over-reliance on anaerobic metabolism relative to an uninfected fish. So what does all of this mean when we're trying to understand how rising temperatures might impact fish? The question is complicated because, of course, being ectotherm's temperature also directly impacts fish behavior and physiology. So first, we need to understand a little bit about the impacts of temperature on fishes and ectotherms more generally. So as I mentioned earlier, pumpkin seeds seem to be able to survive across a really wide temperature range. Again, they're present in places like Spain. They are already in areas where temperatures are really hot. But that made us wonder whether they might have some resilience to warming in our lake sites as well. And so my PhD student Marie collected fish from Lake Cromwell. So this is this really kind of wormy lake. She held them at 1 to 3 temperatures for 3 weeks, and she measured aspects of their metabolism at the whole animal, but also the subcellular level. And the 3 different temperatures we chose corresponded to different things that we see in our system. We had 20 degrees as our late spring temperature. Midsummer temperatures were around 25 degrees. And then we had this kind of 30-degree heat wave, future heat wave scenario. At the moment, during heat waves, we can get water temperatures up to about 28 degrees. So we want to push them a little bit beyond that to really see if they're able to deal with those kind of projected warming conditions in our system. And what she found was that even though the maximum metabolic rate increased with this acclimation temperature, which is what you expect given the way that tend to impact metabolic rates, this wasn't mirrored by an increasing standard metabolic rate or the minimum amount of energy that you need to sustain your functions, or a decreasing in your overall aerobic scope or your ability to engage aerobic behaviors more broadly. And that actually suggests that there's thermal compensation happening, where an organism, like in this case our sunfish, are actually able to maintain and adjust their biochemical and physiological processes to maintain performance across all those temperatures. So that's, I mean, potentially a good thing for the sunfish. They're able actually to cope with these warming temperatures, probably why they're able to deal so well in warmer climates. And one way that they may be able to do that is by using different metabolic pathways. So Marie also measured the activity of different enzymes in the mitochondria and different tissues. And she found that in the gills, which obviously are really important cardiorespiratory organs in fishes, there seems to be a switch from predominantly aerobic to more anaerobic pathways of energy production. And so she looked at the activity of lactate dehydrogenase, again, an important enzyme in anaerobic metabolism, and the activity of enzymes involved in the electron transport system, so really important for energy production in the cellular respiration pathways. And what she found was that at 30 degrees, we have this increase in LDH activity and a corresponding decrease in the electron transport system activity. So again, seems to be this increased reliance on anaerobic pathways at higher temperatures. So how does this thermal compensation happen? Well, one of the ways it can happen is through acclimation. And that's where this is plastic response that occurs within an individual when it experiences a change in temperature over time. Basically, if you expose an organism, an animal, to a higher temperature for a given amount of time, it allows them to then better tolerate warming and maintain the performance at these new temperatures. So they can shift their performance curve. This is just a really stylized performance curve. They can shift their performance curve if they've been exposed to warmer temperatures for a given amount of time. That allows them to better tolerate warmer temperatures in general and maintain performance across a broad range of temperatures. And so this acclimation is a potentially really important mechanism that allow fish to compensate in the face of warming. One of the most common ways to estimate thermal tolerance in ectotherms is using the critical thermal method, specifically CT max, that we use to test tolerance to warm temperatures. You can do similar experiments called CT min, where you test tolerance to cool temperatures as well. These methods are really pretty easy to run. You can test 5 to 10 individuals at a time, so you have a pretty high throughput. The way they work is that you put a bunch of fish into this sort of experimental side of the arena, and you can increase or decrease the water temperature at a constant rate. It tends to be about 0.3 degrees Celsius per minute, but that rate really depends on the species that you're looking at. And what we're doing is looking for a loss of equilibrium. We're looking for the temperature, in this case, in the CT max, the higher temperature that a species can, or an individual can tolerate before it loses equilibrium and is unable to maintain its position in the water body. It is non-lethal. For this test to really work, for us to really consider something as being the critical thermal maximum, it has to be non-lethal. So that's, again, a huge advantage and approach over some other methods to estimate thermal tolerance. And as a result of all these advantages, this approach really has gained in popularity across the years, even though it's quite an old technique. It's back to the 1970s where we start seeing this approach in the literature. But as you can see in this sort of graph here, there's been a real increase in the number of studies on CT max, not just in fishes, but in ectotherms more generally. And that is, I think it's a good thing. I think it's interesting that people are starting to experimentally test thermal tolerance in different organisms. Unfortunately, there's not much standardization or conventions for how these experiments get run or reported. And as a result of that, again, given this growing interest, we recently put together a practical guide for measuring and reporting these kinds of experiments. So if you are interested in critical thermal method as a way of looking at thermal tolerance, I really encourage you to read this paper and have a look at it. Hopefully it gives you some good tips and tricks for how to best run your experiments and potential pitfalls that you can avoid that we fell into, and hopefully you can learn from our mistakes. The other nice thing about CT max experiments is that it's a metric that can be used and compared across different life stages. This is a project that was led by colleague Zara Cohen, where we developed a method for estimating similar behavioral CT max endpoints in different life stages, so from adults, but also embryos and larval fishes. This is my head here in this publication. It's my claim to fame of having my back of my head in a scientific publication. And this, again, is really interesting because it lets us start answering questions about, well, what life stages might be most vulnerable to warning? And we've started looking at this across a bunch of different species. What we've been able to show is that embryonic stages are actually more resilient than was previously thought to these kind of warming temperatures. And again, so because we have this method we can compare across life stages, it provides us an opportunity to really look at this kind of more broadly. And these are data that are part of a larger project also looking to better understand thermal tolerance across different stages of embryonic development. So hopefully we'll have some results on that to present pretty soon. So acclimation might provide some protection from warming. We can measure warming tolerance across different life stages. But how long does it take for an individual to acclimate after they've been exposed to higher temperatures? Do they need to be in warmer waters for weeks before they gain some resilience to survive in a heat wave? How dynamic are these changes? The answers are we don't really know. There's actually not that many studies that have looked to see how fast an animal can acclimate once it's exposed to a warmer temperature. And the terminology is also a little bit confused in the literature. So to better quantify acclimation dynamics, my student Jeremy went to Sweden to the Kristineberg Marine Research Station to study a few different species of common small-bodied fishes. For people who have been falling asleep, this is the marine part of the talk, so you can wake up now. I'm going to talk about marine species for a little bit. What Jeremy did here in Kristineberg was to define a few metrics that we can use to easily compare species or populations from different habitats to understand how fast or how much they can acclimate. So I'll just define a couple terms because, again, I think this is really important for our understanding. We can talk about acclimation response ratio, and that's the magnitude of change of the initial CT max following an increase in temperature. So if you have an acclimation response ratio of 1, it means that for every 1 degree increase in your acclimation temperature, you would expect you have a CT max that would increase also by 1 degree Celsius. So that's kind of what we consider like a perfect acclimation response. You can also look at the response induction time, and that's measured as how the duration of exposure to a higher temperature that you need before you get an observable change in CT max compared to the individuals that are in the control temperature. So how much time do you need? A couple hours, you need a few days. How much time do you need to be exposed to a warmer temperature before you start getting a noticeable increase in your CT max? And lastly, we can talk about the time to full acclimation. That's the exposure duration you need to complete your acclimation. That is the time you need before your thermal performance curve to shift so that you're achieving the sort of maximal performance of that new temperature, when more time in that temperature isn't going to increase your performance any longer, where you've kind of reaped all of the benefits of that acclimation temperature. So we sort of defined some of these terms, and we wanted to explore those acclimation dynamics across different species. And what Jeremy found was that there is actually differences in these dynamics across species. What he did was expose fish to either no acclimation or either 3, 6, 24 hours or 4 and 10 days to 5 degrees above ambient temperature, and then look to see again how the CT max changed based on these exposure times. And what he found was for flounder, this was on a flounder species, we had the T induction about after 4 days, as well as T steady. So here you can really see this sort of change where we first start seeing a noticeable increase in CT max occurs at about 4 days. But then after 4 days, even if you're exposed longer to 10 days at that higher temperature, you're not gaining anything in CT max. So T induction is also around that day mark, that 4 day mark. It was different for wrasse species, Goldstony wrasse that we tested. In wrasse, we had our T induction time that happened within 6 hours, so pretty fast compared to flounder. But they actually, even after 10 days of exposure, didn't quite reach T steady. They were still gaining CT max relative to the previous exposure duration. So we didn't actually reach T steady in our experiment in this species. In stickleback, we had this T induction that was even faster. It was within 3 hours we started seeing already a noticeable gain in that CT max. And T steady was reached after about 4 days in this species. So again, some of these dynamics do seem to be quite species specific. He also tested whether these dynamics are similar across life stages by comparing adult and juvenile zebrafish with collaborators in Norway. And he found very similar dynamics across life stages. We have our T induction happening around 3 hours, so also really fast, and T steady occurring about 4 days, regardless of whether you're testing juveniles or adults. Interestingly, this acclimation capacity does seem to differ among species. We found the highest acclimation response ratio in our wrasses, a seemingly lower response ratio in these shallow marine species like the stickleback and the flounder. Maybe they're living closer to the thermal maximum. They can't fully compensate. Although these species did seem to acclimate faster. They had this lower T induction time. So there's really interesting trends here that are only really beginning to be explored and I think should be studied more in other species. Now let's try to tie all this together. What about the parasites? Well, we understand something about how fishes acclimate to warming waters under the best of circumstances, but are infected fish more sensitive to warming? Again, like we are looking at this in the absence of this really important biotic interaction. And Jeremy wanted to this time look at sunfish from our Lake Cromwell across that natural infection gradient and acclimate these fish to, again, our 3 temperatures, 20, 25, and 30 degrees Celsius, and then measure their CT max to see whether or not there were any relationships with any of the infections that we typically observe. And what he found was that sunfish with 1,000 black spots had an estimated 0.5 degree CT max lower compared to fish with no black spots. That might not sound like much, but it actually is. Again, we have some of our fish with black spots that range in the 2,000 to 3,000 range. That's about a degree to 1.5 degrees difference in CT max in those really infected fish. Again, it is a lot when you think about the differences that we typically see in these metrics across individuals and species. But not only that, he found that survival in the days following a CT max test decreased by about 25% in this warm acclimated infected fish, and especially fish that were infected with these yellow grubs, these big chunky parasites that really tend to be present around the heart and the gills, was really responsible for death in our fishes following this heat event. And so it really suggests that there might be this increased survival cost to incurring some sort of heat damage in our infected fish, and this increased sensitivity to acute warming in fishes that are infected. So wormy fish seem to have a harder time reaching their full thermal compensation and maybe also accumulating extra thermal damage during these rapid temperature changes that they might be experiencing during a heat wave. But what about their acclimation dynamics? Do different exposures to infection impact the ability of our fishes to acclimate? My master's student, Andrea, took advantage of this lake system that I presented earlier to explore whether acclimation metrics differ across lakes with different infection prevalences and intensities. So she collected fish from our Lake Triton, where there is no infection, Lake Crush, where we have intermediate levels of infection, and Lake Cromwell that have higher levels of infection, both prevalence and intensity, to really look and see whether or not there were differences in these dynamic acclimation metrics that I mentioned before when fish are exposed to water 5 degrees above ambient. And what she found was that compared to control fish that were kept in ambient temperatures across our 60-day experimental treatment, she found that CT max actually increased for all lakes after 60 days in warm acclimation. Interestingly, the CT max of the fish in our Lake Triton with no infection was the highest out of all of our lake populations. So the lake with no natural parasite infections had a significantly higher CT max than all the other 2 populations that we studied. In terms of acclimation dynamics, she found that T induction and T steady were very similar across the 3 populations, about 3 hours and 10 days, and that the acclimation response ratio was also similar at around 0.5 for all lakes. But again, all of these exposure durations, the infected Lake Triton had higher CT max than the other 2 populations. And interestingly, Lake Cromwell, our most infected lake, also seemed to reach its performance at around 10 days, but then it's doing something weird. It's like dropping off around 21 days and at 60 days warm exposure, which might suggest that long exposure to these high temperatures might be more difficult for them to deal with. Again, perhaps they're accumulating some sort of thermal damage. We really need to explore this a little bit more. It was unexpected and quite an interesting result that we need to begin. But surprisingly, she actually didn't find any relationship. If we're looking closer at the lakes where there is infection, we didn't see any clear trends with the intensity of infection across any of our acclimation durations that we tested. So even though Lake Triton had a higher CT max in both of the infected lakes, there didn't seem to be any strong relationship between CT max and the number of either black spots or trematide parasites, black spot or tapeworm parasites that a fish had. So there really wasn't any strong relation to any of the acclimation durations with the intensity of infection. So again, a surprising result. So I'm noticing the time. I'm going to try to wrap up here. And I think my message here for all of you is that I think we all should be thinking more about parasites. They're often ignored by experimental biologists and biologists in general. I don't think they should be. Host-parasite relationships are pretty complicated, but I think they're really, really interesting. Parasites can impact morphology, physiology, and behavior of their hosts. I do think it's worth studying and thinking about, especially in the context of rising temperatures and heat waves. And again, we're only really just starting to do these experiments where we're looking at a combination of these different factors. I think integrating parasites into studies of other global stressors as well can be especially revealing. So if you work on not temperature, but hypoxia, toxicants, microplastic, anything like that, I really think that there's value in including parasites in your research. And I'm just going to hopefully take a couple more minutes just touching on the theme of the seminar season series, which is reimagining the future of oceans and fisheries. I presented work on sunfish. Of course, I think it's a great model species. But of course, I think a lot of what I talked about today can be generalized to other species of more commercial interest or fisheries. In the lab, we also are looking in projects. We're developing more and more systems on American and European eel, also smallmouth bass. So I do think that some of the things I talked about are relevant for fisheries. And I'm going to rephrase this question a little bit slightly for reimagining the future of oceans and fisheries to how can we best prepare for a potentially warmer and warmier world? And as a mostly empirical biologist, I think I've thought a lot about the challenges associated with designing experimental and observational studies in a way that's not just scientifically interesting, but also useful to managers who are trying to design policy to help answer these questions and are really trying to be good stewards of our oceans and our fisheries. And I think sometimes there's at least a perceived tension in terms of what the best approach is for an empiricist. I mean, do you do a bunch of lab experiments to better establish cause and effect, or do you do a field study where you prioritize ecological reality, but maybe less control on the environment? And I guess I've tried to show you with these different aspects of my research program is that I don't think it has to be an either or approach. I think policymaking in this new future requires a diversity of approaches and clever combinations of lab and field-based research to address these pressing challenges. And this is something that colleagues and I just wrote in a perspective piece for conservation physiology, where we try to give our thoughts on how to collaborate across disciplines and sectors to achieve policy and conservation goals. And again, how working across this lab field continuum can really be kind of a bright spot in terms of imagining the future of different fisheries, different oceans. Not just, again, I think this is very broadly applicable, but how we can kind of work in tandem to help make better science for policy and practice. Sometimes it's hard. I think even if we're all rowing in the same direction, not everyone agrees exactly on the best path forward. Sometimes there's really heated debate, and especially these days, given some of the current political realities going on, there's often a lack of trust in terms of science and scientific research. And that makes it difficult to achieve consensus and convince policymakers that what research we're doing is valuable. And so, again, I think it's really important that we work collaboratively, that we have integration across disciplines, that we really get people who have expertise in different areas to work together to foster collaborative environments, and also transparency and reproducibility in our research work. And again, with some collaborators, we wrote a commentary for Journal of Experimental Biology a couple of years ago, where we tried to give some ideas about how we can achieve better consensus and foster transparency and reproducibility in our research. That can involve a few different things. Sharing data is really critical because it helps others understand what you did, allows for reanalyses and comparisons with new data in the future. And again, I really think that's important for moving things forward. Open materials, also being really clear and honest and open about what you did. We've done a lot of work. I mentioned this methods paper that we published about CT Max experiments. I mean, I think those things are great. We try to provide some tools for open reporting. What should you describe when you're reporting experiments? So it's easier for people to understand what you've done and help replicate your research. And again, give people more trust in what you've done. And also pre-registration, which is something that is not very common in our field, I would say. It's very common in some fields, like in psychology, for example, where you pre-register your experimental design, you describe what you're going to be doing, how you're going to be doing it, what your hypotheses are. Your paper basically gets accepted before you have any results. Then you go out, do your experiments, and publish the results, regardless of the way that you said you were going to do, regardless of what you find. So it tends to be more resilient to some of the problems that plague statistical analyses in a lot of ecological and experimental sciences. Another thing that I think we need to do is, in this time of global change, is bring together scientists from different parts of the globe through global initiatives. And one of those is something called the Thermal Ecology Alliance. It's just been announced. It's a global collaborative initiative that's bringing together scientists who are interested in looking at the effects and the impacts of extreme temperature events on biodiversity. It's really aquatic focused, but again, the Alliance is looking to expand. This is an initiative by Patrice Potier and colleagues. If you're interested in thermal ecology, please go and check them out. Join the initiative. My lab's part of it, but we're looking for collaborators from across different fields and with different expertise in terms of systems and questions as well. So please go check it out. It's really great. Also, my last plug before I wrap out, please come to our meeting. Aslo Sil is hosting a meeting in Montreal in May. I'm organizing a session called To and From Aquaculture and Fisheries in a Changing World with Jeremy de Bonneville and Sophia Savag, two of my former students. So please, if you're interested in these topics and you want to discuss more, I really encourage you to submit your session. Come to Montreal in May. It's a great place to be. And if that's too far for you, well, then come to the ICBF meeting in Vancouver in July. I'm also organizing a session there with some other students called Bridging Experimental Biology and Conservation from Mechanisms Application. So again, if you are interested in exchanging on any of these ideas, please come and join us. It would be really great to see you there. And with that, thank you to my collaborators, my fantastic students. I tried to give a nod out to them where I could, but this work wouldn't have been possible without a whole bunch of people who were just really excellent colleagues and good friends. And thank you again for the invitation.
Thanks very much, Sandra. And I think we've got time for a few questions. There's one in the chat which is a little bit close to my heart, but the question is from Ben Nyman and is asking, do you have any current work or plans to explore less visible parasites through 18S or other molecular methods?
Yeah, that's a really great idea. It's a really something we're really trying to do. So I didn't mention any of the work that I'm doing on American eels, but we are working in collaboration with the Quebec government on doing some work on American eels. They're super important fishes, both commercially and culturally, but they're also really interesting because they have recently experienced invasion by a nematode from probably from Europe, but originally from Asia. It's a swim bladder nematode. It's a big problem in Europe, so we collaborate with our European partners also looking at the impacts of this invasion in Europe as well on European eels. But it's a growing problem in North America. We're tracking its spread through the St. Lawrence estuary system, and basically it's really hard to detect. And so yes, we're trying to find ways, better ways through PCR and other molecular methods to be able to tell when an individual is infected, because that's the biggest thing that's hampering a lot of work on this species. Like we can go out and collect a bunch of eels and do a bunch of experiments on them, but we have no idea whether they're infected or not. And so it means that we need to have really high sample sizes to get anything useful. And when you're working with kind of a vulnerable species, that makes it very, very challenging. So I don't have very much expertise in molecular methods. I have collaborators who do, and we're collaborating with vets also. One thing that we're trying to do, which I think is really cool, not a molecular method, but is using techniques in X-ray and ultrasound. So we did an experiment with some vet collaborators using ultrasound to try to detect infection in these less visible infections. It wasn't a complete failure, but it was a pretty big failure. Let's put it that way. So ultrasound doesn't seem to be the way to go. But X-ray may be, maybe something that is promising. And so there's more and more these portable X-ray machines that you can bring to the field, sort of scan a fish quick and look for signs of infection in bladder or other parts of the body, places like the liver. So we are exploring that a little bit more, hopefully.
So we're coming to the end here. We've got maybe a few more minutes for quick answers for the next question. So maybe we'll take one from Murdoch, who, if you could unmute yourself, and then if there's also a reminder, if there's any questions from room 107, maybe we'll go to you next, and then I guess we'll probably quickly wrap up after that.
Yeah, thanks for your really fascinating talk. I had a question about your observation of really large variation in spatial rates of infection, but also another thing is your theme of your talk is like wormier world. So what you should also be looking at is like trends or changes in interannual variation, anecdotal information suggests that there can be large interannual variation in rates of parasitism in lakes. I do a lot of trout fishing. I noticed actually, I'm really surprised. Some years I don't see any, and other years the same lake, there's same fish, there's a lot of worms and so on. So what are the hypotheses and what are your investigations showing about factors in these little geographic areas that impact spatial variation, parasitism versus interannual variation in the same waters? Thanks.
Yeah, that's a great question, and one that we have data on and we haven't analyzed yet, in the sense that, as I mentioned, we have these 3 lakes. We have more, but we've really focused on these 3 lakes because I think they're interesting for the just diversity and the parasites that they have. But every 2 weeks, starting in June and going towards September, October, we kind of do these. We have these sort of permanent transects, and we look at prevalence of infection. So we count the number of fish that have these black spot. Again, the black spot is a really, really cool model for us because we can go and non-invasively sample the fish and look at prevalence. And that's exactly what we hope to be able to do, is sort of every 2 weeks or so, since 2019, we've got data now where on these permanent transects, we have the prevalence of infection across different size classes in our pumpkin seed and other species, but mostly pumpkin seed, what we find here. And that's what we hope to do in the future, once we get enough years of data, is to look at interannual variation, but also across the season, how these things are changing, at least as far as the prevalence of black spot goes. As far as intensity goes, we do have some data, and like you said, anyone who kind of goes out and just out fishes for any species, you do notice that there are some times of the year where you're going to get more wormy fish than others, notably, kind of warmer months in the summertime. And so, yeah, those are super interesting questions. And again, we've got some data that we're hopefully going to be able to explore more detail in the future. I know that's not a very satisfactory answer, but it's a really interesting question.
I'll read out the question for you. It says, "Great talk, thank you. I'm curious about the interconnectivity of the lakes in light of the varied infection levels seen in them. Are the more infected lakes downstream of the less infected lakes, for example? How do you think parasites are being transported between the systems, if at all?"
Yeah, I think that's a great question. I wonder if I can, sorry, I hate it when people scroll back 50 million slides. But I think it's worth kind of having a look at oh, here we go. You can look at this map, I guess. Okay, yeah. So yes, these lakes are interconnected. You can see here, there's this little, so Triton is actually head lake. So that goes to this idea about parasites being transported downstream. I think that the question was getting at. Flows into Lake Crash here, this sort of intermediate lake, and then that goes down to Lake Cromwell, our higher infected lake. So I do think that there is some of that, Lake Crush. I mean, it receives, this is kind of one of the larger lakes in the area. It receives input from other lake systems around it as well. That said, it's not the sort of connectivity in terms of the hydrologic flow is not the only thing that's important here. The thing that's really important, especially when you have these parasites that aren't directly transmitted, that are actually transmitted through these intermediate and final hosts, is how the hosts, all of those hosts, are moving through the system. And one of the big advantages that trematodes, these black spot trematodes, have is that their final host is a bird, and birds can get around everywhere. So we have these birds that are kind of present across all of the lakes. These lakes aren't actually, you can see here the sort of spatial scale. They're not that far apart. The home range of a heron or something is more than large enough for them to access all of these 3 lakes. And so it really, like allows the parasite to be distributed kind of broadly across the landscape, including upstream. So they can kind of get around this hydrological constraint associated with the flow of water downstream. Now, why is it that Lake Piton still doesn't have parasites? This is the question. The herons could get there. Why is it? I mean, that I think really has more to do with the bathymetry of the lake, the shape of the lake. Like, this is a very small lake. It doesn't have a whole lot. It falls off very steep. It's not particularly deep, but it falls off very steep on the edges. And it actually doesn't have very good habitat for another important species in this whole system, which is the snails. So there's just it's not a very good habitat for these macroinvertebrates that are a key part of the life cycle. They don't have snails. If the snails aren't there, you're not going to get the parasite. So in some ways, that's where these parasite host dynamics are super interesting, because it really is a community level question that you have to sort of tackle. And same thing when thinking about this in terms of a warming world and making predictions about what's going to happen in the future. There's so many moving parts, and the parasites is true. Like, they get an advantage to some extent, in the sense that they develop faster in the host when the temperatures are warmer. They're ectotherms too. They develop faster in the host when the temperatures are warmer. It potentially means they have more reproductive cycles within a season as our warm season is expanded. But they are still reliant on all of these different species. And if all of those different species are being differentially impacted by temperatures, maybe our fish are quite resilient to the warmer temperatures. But if the snails are not, that's still going to impact them. So these things can get very complex really, really fast, and which is why I think it makes them really, really interesting. So. Sorry, I have a tendency to go off I get very excited about these things. So my answers are sometimes long. I hope I do answer your question, though.
You gotta thank you in the chat.
Okay.
Yeah, and I think that's a great answer, and it's also a good lesson that you need to remember all the different life stages when you're studying these parasites. It's part of what makes studying them so fun, I guess. I'm gonna wrap this up. So I'd just like to thank you on behalf of the Institute for the Oceans and Fisheries for excellent presentation, Sandra. It's super interesting for me. There were lots of take homes, and as someone who works in disease but not in the physiology side of things, I was learning a lot as well. So I really appreciate you sharing all your work.